Warm greetings to all. I am the Karthika, PhD scholar, working under the guidance of Dr. Velmurugan, scientist, KMCH Research Foundation, and under the co-guidance of Dr. Shanker, Assistant Professor from School of Biochemical and Biotechnology at SASTRA University. My PhD title is on profiling of gut microbiota and its metabolites during endocrine-disrupting chemical-induced glucose dyshomeostasis, implication of host-glucose metabolism. Over the past few decades, diseases have changed worldwide. Non-communicable diseases like type 2 diabetes are now very common. At the same time, exposure to the chemicals has increased due to the industrialization and the modern lifestyles. Chemicals can disturb the hormones, but how they cause disease like diabetes is still fully not clear. The new study suggests that the gut microbiome may play a major role in this progress. There's a problem statement. The chronic low dose exposure to the endocrine disturbing chemicals may contribute to the diabetes via the gut microbiota alterations. To investigate, we have adopted an integrated approach combining the meta-analysis in vivo mice model and the fecal microbiota transplantation experiments. We also simulated the real-world chemical exposure by administering the mixtures of multiple chemicals at environmentally relevant dose. To better understand the metabolic effects that the chemical exposed mice were compared with the high-fat with high sucrose diet models, both the groups developed the glucose intolerance, but only the high-fat group showed the obesity. In contrast, the chemical exposure resulted in the distinct non-obese diabetic phenotype. The chemical exposure can cause the microbiota changes in the microbiome known as dysbiosis. There was the reduction in the beneficial bacteria, which helpful for the production of short-chain fatty acids, along with the decreased balance between the firmicutes and the bacteria. At the same time, the harmful bacterial groups such as proteobacteria, bacteria, and especially the sulfobacteriota was increased. It is known to produce the hydrogen sulfide, and the excess of hydrogen sulfide, which could lead to the cell damage, weaken the intestinal barrier, and increase the inflammation and disrupt the metabolism. Interestingly, the pattern was different from what was seen in the high-fat diet model, suggesting that the chemical-related diabetes may develop through a different microbial mechanism. These findings highlight the potential of microbiota signatures, particularly the sulfobacteriota and the microbial hydrogen sulfide, as a novel biomarker for disease reduction and risk stratification. In conclusion, the study demonstrates that the chemical exposure can reshape the microbiota and drive the non-obese diabetes through the microbiota-mediated mechanisms, may opening a new avenue for the precision medicine. The future therapeutic strategies in the gut microbiome research focus on restoring the beneficial bacteria to treat the disease. The key approach that the sulfobacteriota-related stress markers and inhibit the sulfate-producing bacteria, and that could lead to the microbiota targeted therapeutics. I need to acknowledgement for the funding agencies for the financial support, and I need to deeply extend my thanks to team members of KMCH Research Foundation and the supporters. Thank you.